Hello everyone, and today we will be talking about insulin function. So in this video we will talk about insulin functions, including its signaling pathway and how it acts, its different pathways, how it is released. So this is going to be particularly helpful for those who are giving Usmle step 1, because there's a lot of mechanisms that we're going to talk about. So without further delay, let's just start the lecture. So first, we need to know the insulin, where it is released from. So it is released from pancreas, and this is the organ that I drew here is the pancreas. The pancreas is very close to the duodenum, so it's the secretions of the pancreas actually goes into the duodenum to digest the food that a person eats. Now, the pancreas is of two types. One is the exocrine pancreas, and one is the endocrine pancreas. So the exocrine pancreas has a lot of the ducts, like the ductal secretions. If you see here, they have this acini. These are the ductal cells. So this ductal part is called the exocrine pancreas, and they release the amylases, lipases, all of the things, and also alkaline secretion, all the things that help in the digestion of the food. So they're going to come here and are secreted into the duodenum. And then another is the endocrine portion. Endocrine portion is the one that actually empties or releases the insulin into the blood directly. So these are the islets of Langerhans cells. So remember, this is the islets of Langerhans cells. These represent only 1% of the pancreas. 99% is exocrine or acini. So now we have the islets of Langerhans cells here, and in the center we have the beta cells, and then we also have the alpha cells. These are the two most important cells in the pancreas. So now we have the alpha cell and the beta cell. The beta cells are very important. They are the ones that release the insulin. So insulin is released by this. Also, the alpha cells produce glucagon. So glucagon has A in it, glucagon. So that's how you can remember that it is released from the alpha cell. Now, the insulin is released in response to what? In response to hyperglycemia, increased blood sugar levels. So now if you see this duodenum here, the food that you eat is going to go down the stomach and into the duodenum. Once it's in the duodenum, it is going to be the sugar levels and all the sugar that you eat in your food, it's going to be absorbed from the GI tract into the blood. And once in the blood, it's going to cause increased blood sugar levels. Now, this increased blood sugar level is going to stimulate the insulin release, while glucagon is released when you are fasting, when you have decreased glucose levels, because it increases the blood glucose level. Insulin is released when you are in the fat state, when you have increased glucose levels in the blood, because it decreases the glucose levels normally. Now, let's say someone ate, and now let's see how the insulin is released from the pancreas. So now let's say we are just magnifying one of the beta cells here. This is the beta cell that we are seeing from the islets of Langerhans cells. Now, in the beta cells, first let's talk about insulin. Remember, insulin is a protein hormone. It is first synthesized as a proinsulin, and then it's converted into insulin plus C-peptide. So now let's say this is the DNA and this is the gene for the insulin secretion. So we transcribe it into messenger RNA. It's going to come out, and then it is going to be on the rough endoplasmic reticulum, which we have the ribosomes, is going to be synthesized as a protein, as a proinsulin, and then it's going to be packaged into the Golgi apparatus, and then into the Golgi apparatus it's going to be cleaved into insulin and C-peptide. So if you look down here, this is the insulin structure. So insulin has two chains that are bound by the disulfide bonds. So this is one chain, this is another chain, and they are bound by this yellow chain, and that is the C-peptide. So in the packaging, in the vesicle, this chain, this entire insulin is actually broken down into this one structure of C-peptide and this one structure of insulin. So now we have the insulin and we have the C-peptide here. C-peptide here is very, very important, which we'll talk about in a bit, why C-peptide is important. Because remember, C-peptide represents that you are actually making endogenous insulin. So when someone has high C-peptide levels, that means that the insulin is endogenous. If you have low C-peptide, it means that you are taking insulin from outside. It's exogenous insulin. So there is a drug called sulfonylurea that also increases endogenous insulin. It releases endogenous insulin, used in patients with diabetes, and that also will cause increased C-peptide levels. Now let's see how insulin is released. So first, we need to know that there are different types of glucose transporters. Remember, glucose cannot just enter by diffusion into the cell, so it needs a transporter. The transporter in the pancreas is the GLUT2 transporter. Remember, the GLUT2 is important. So now, GLUT2 is, why is it important? Is the reason is that because this is, first of all, it is insulin independent, so it does not require insulin for uptake of glucose. So remember, most tissues require insulin to uptake their glucose, but there are certain organs that do not. For example, the pancreas and also the liver. The liver also have GLUT2 transporters. Also, those organs which have GLUT1 transporter also do not require insulin. For example, RBC, for example, brain, they do not require insulin. So GLUT4 are the transporters we will talk about. Those are the ones that require insulin for uptake of glucose. Now here, GLUT2 does not require insulin. So now, once you eat, you have increased glucose in the blood. This glucose is going to enter directly into the pancreatic beta cell. Now, what it inserts into the beta cell. Now, another thing about the GLUT2 is that you can remember GLUT2. There's a bidirectional flow, like two flows, which means that this glucose can enter, but it also can leave through the GLUT2. Other transporters don't do that. Other transporters, you can only get the glucose in. Now, let's talk about, so now this glucose enters into the cell. Now, once it enters into the cells, it is a 6-carbon structure. Through glycolysis, it is divided into two 3-carbon structures. This 3-carbon structure is then further divided into 2-carbon structure. And this 2-carbon structure is acetyl-CoA. It's going to go into the mitochondria. This is the mitochondria, undergo Krebs cycle and produce a ton of NADH and FADH, and that's going to go to the ETC, electron transport chain, and generates a lot of ATP. So with each molecule of glucose, you have 2 cycles of the Krebs cycle, 2 Krebs cycles for one glucose molecule. It's a 6-carbon structure. Eventually, it's broken down into 2 acetyl-CoA. So these acetyl-CoA are going to generate a lot of ATP in the electron transport chain. Through the Krebs cycle, they form NADH, FADH. These go to the electron transport chain, produce a ton of ATPs. So once this glucose is broken down, you have increased ATP to ADP ratio. Now, we have a channel here, and these are in the beta cell, and these are the potassium channels. What it means is that there's constant outflow of potassium through these channels. Now, this hyperpolarizes the membrane, so the membrane doesn't depolarize. So you have decreased intracellular positive charge. Now, once you have you're in the fat state, you have a lot of glucose that enters through the cells and produce a lot of ADP. You have increased ATP to ADP ratio. It's gonna put a block here on these channels. Once it blocks these channels, you have a lot of potassium inside the cell. There's a positive membrane potential, and this is going to activate voltage-gated calcium channel. Now, once these voltage-gated calcium channels are activated or opens, this vesicle that we talked about, which contains the insulin and also the C-peptide, is going to fuse with the membrane here. It's going to fuse with the membrane and going to release the C-peptide plus insulin. Now, this C-peptide, insulin, and also remember, amylin is also released with it. In the vesicles, it is packaged with the insulin and C-peptide. So these things are going to come here. Now, once they are here, this insulin is going to go into the blood, and it's going to go to different organs. For example, it's going to come here into the liver. So now, in this cell, on this cell, we are going to talk about the insulin pathway. Remember, insulin is as we talked about, it is synthesized as a protein hormone, right? So it's a protein hormone. Remember, whenever something is protein, they cannot cross the membrane. They need a receptor on the cell membrane to act. So if I draw just a small cell to kind of describe this, so we have cytoplasm, we have the nucleus, we have the cell membrane. If you have a fat-soluble hormone, it's going to directly cross the membrane, binds to its cytoplasmic or nucleus receptor, and bring about its action. If something is not fat soluble, for example, insulin, which is a protein, it should have a surface receptor. Here, insulin is not fat soluble, so it should have a cell membrane receptor. So now, this membrane receptor is a tyrosine kinase receptor. It's a tetramer, remember. it's a tetrameric receptor. Now, once insulin binds to this, remember, this is going to occur in the fats, in the muscles, in the liver, anywhere the insulin acts. It's going to act through this receptor. So it's a tyrosine kinase. Once let's say this insulin binds here, it's going to autophosphorylate the receptor. Once it's autophosphorylate, there is a substrate called insulin related substrate, IRS1. It's also going to get phosphorylated. Eventually, this IRS is going to go through down two pathways. One is the PI3K pathway. The other pathway is the MAP kinase pathway, a ROS MAP kinase pathway. So now, this PI3K is going to go and increase DNA expression, gene expression of some transporters called GLUT4 transporters we will talk about. So they will increase in all the cells where the insulin acts. They will increase the GLUT4 transporters, and this GLUT4 is insulin dependent. It's going to uptake the glucose from the blood under the influence of under the influence of insulin, because insulin binds to this receptor, brings about increased synthesis of GLUT4 inserted into the cell membrane, and then you have increased uptake from the blood. So your blood sugar levels go down, which is the goal of insulin, is to bring it down. Now, the other pathway is not that important, but still just know there is also a ROS MAP kinase pathway. It helps in cell cycle proliferation, regulation, and cell growth. Now, so let's come down here. So it's gonna, remember, the same receptor is here for insulin. Insulin has tetramer receptor. It's gonna phosphorylate here. It's a tyrosine kinase receptor. Again, it's gonna be RRS, and then ROS MAP kinase pathway and PI3K pathway is gonna be activated. Now, once you have increased glucose in the blood, this glucose is gonna, without requiring insulin, because these are GLUT2, enter into the liver. Once it's in the liver, this PI3K pathway is going to activate glycolysis. Once it activates glycolysis, it's going to go down the same pathway that we talked about. It's going to cause increased ATP generation. Another thing that, so it activates glycolysis is the first action of insulin in the liver. Any organ, it can cause glycolysis. Second is it also increases, it's going to cause this glucose to link to each other and form glycogen, from glycogen storage. So remember, increase, so far these are the two actions you need to know for insulin. Increase glycolysis, increase glycogenesis, which means formation of glycogen, which are stores of glucose. Now let's go down to this. So we have then this insulin is going to come, and they can come to this here into the muscle as well. So in the muscle, it's going to again come and has this own receptor, the tyrosine kinase receptor, phosphorylates, IRS phosphorylates, and there is PI3K and ROS MAP kinase pathway. This PI3 is going to increase the expression of which GLUT transporters we talked about, GLUT4. So insulin is going to insert GLUT4 receptor here, and GLUT4, remember, are insulin dependent. It requires insulin for uptake of glucose. If you don't have insulin, they're not going to uptake the glucose. Now, this glucose in the blood that you ate after in the fat state, it's going to go and enter into the muscle. So now this is going to, again, just like liver, it's going to be broken down into and generate. We can generate a lot of ATP from it. Also, it's going to broken down, and also it can actually connect and form glycogen, muscle glycogen. Remember, like I said, GLUT2 also has bidirectional flow of glucose, which means that, for example, if someone is fasting, this glycogen can be broken down, and it can go through the GLUT4 and released into the blood in low glucose state, like fasting. GLUT4 cannot do this. So, for example, muscle glycogen cannot be broken down and released into the cell. So now next up is this is the fat cell. So in the fat cell, again, insulin is going to come, bind to its tetramer tyrosine kinase receptor, phosphorylate itself, IRS is phosphorylated, and then it's going to cause PI3K MAP kinase pathway activation. It's going to go to the DNA gene expression of which glucotransporters? GLUT4. So again, here you have increased GLUT4. GLUT4, again, insulin dependent. It's going to uptake this glucose. So now let's see what it does in the liver. So in the adipose tissue. So now in the adipose or fat cell, this is going to, once this glucose is in, it's going to break down, just like a simple glycolysis. It's break down into 3 carbon structures. This 6 carbon structures is broken down to 3 carbon structures. One is dihydroxyacetone phosphate, and one is the one is this glyceraldehyde 3 phosphate. This glyceraldehyde 3 phosphate is broken down further, just like in normal glycolysis, in acetyl CoA, which is 2-carbon structure. And this dihydroxystone phosphate, remember, in the fats we have a lot of triacylglycerides. This is let's say the glyceride, and these 3 are the fatty acids. 3 fatty acids attached to it. So these are the triglycerides. Now this dihydroxystone phosphate is going to be converted by the under the influence of insulin into glycerol, which is the head of the triacylglyceride. While the acetylcholine, the insulin is going to cause it to form fatty acids, and these are the 3. These are the 3 fatty acids that are attached to glycerol. So these 3 are going to come here, attach to the glycerol, form and form triglycerides. So remember, the actions here so far: increase glycolysis, increase glycogenolysis. Third is increased TG synthesis, or fat synthesis. So far it's an anabolic hormone, which means it synthesizes things rather than break down things. Apart from glycolysis, it does increase glycolysis. Now, if you have increased TGs in the muscle, it also does something else. For example, it increases the uptake of amino acid. When you eat, you have a lot of amino acids in the blood, and it's going to uptake that and help in protein synthesis. So the last thing is that it also increases protein synthesis. So these are the general things that the insulin performs. So hope this video was helpful. It was just a short video just to tell you how insulin acts and what are its different functions, how it brings down the glucose, what are its eventual goals. Remember, it's an anabolic hormone, increases glycogen synthesis, increases glycolysis, increases TG synthesis, increases protein synthesis. So hope this video was helpful. So if anyone requires tutoring, they can email me here. Thank you for watching.